Hello children, hope you are doing great. This is a season to read and I'm pretty sure that you are doing it to the fullest. Today we'll be discussing a chapter called Plant Kingdom. Important chapter because the weightage for this chapter is approximately 3 to 4 questions. Without wasting any time, we'll get into the thick of this chapter. Now, if you talk about the NCERT textbook lines, I want to go according to the same lines. Now look here. Fungi, bacteria, alga. In Whitaker's classification, where they are kept. In Linnaeus classification, fungi are kept under kingdom Plantae, whereas in Whitaker's classification, it is given a separate kingdom, fungi. Coming to bacteria, in Linnaeus classification, it is kept under kingdom Plantae because it contains cell wall, whereas in Whitaker's classification, because bacteria are prokaryotic, it is separated into a kingdom called Monera. Coming to alga, this is important. There are 3 kingdoms, all right. In Whitaker's classification, this alga are kept. If you look at blue-green alga, it is kept under Monera. Diatoms, dinoflagellates, golden alga, chlorella, chlamydomonas, they are kept under kingdom Protista, and the remaining alga are kept under kingdom Plantae. This is important. Now, without wasting any time, we'll talk about the different types of classifications. 3 types of classifications, children, we'll be discussing today. Criteria, example. So these are the side headings we take. First, artificial system of classification. What is the criteria? Few morphological characters. That is the criteria. And what are the examples for that? First, Linnaeus sexual system of classification. Remember that. Here he classified flowering plants into different groups based on the number of stamens or nature of androecium. Because only one character is taken, so therefore it is rightly called artificial. Even Aristotle classified plants into herbs, shrubs and trees based on one character, nature of stem. You can even call that as artificial. Annuals, biennials, perennials, xerophytes, hydrophytes, mesophytes. These are all examples for artificial system of classification because you have taken only 1 or 2 criteria, especially morphological characters. Your textbook says equal weightage is given to both vegetative and reproductive characters in this artificial classification, which is absolutely right. Now, we have got 2, Linnaeus classification and Aristotle's classification come under equal weightage, all right, to both vegetative and sexual characters. That's about artificial. Then we talk about the next type of classification, that is natural. Look here. If we classify plants or animals into groups based on all the branches available for us, not only morphology, anatomy, biochemistry, physiology, embryology. If you take all the branches as a criteria for classifying plants, then the natural affinities are taken into criteria and the plants in that particular group resemble, all right, greatly. Such type of classification is natural system of classification, and an important example for that is the Bentham and Hooker classification. In all the Commonwealth countries, including India, the herbarium sheets and are kept in accordance, in order of Bentham and Hooker classification. It is widely followed classification, you can even say that. Then, phylogenetic system of classification. It is also based on all the morphological characters, information from different branches of science. But evolutionary tree or evolution also is taken as a criteria. Means the organisms belonging to the same taxa have a common ancestor. Say for example, lion, tiger, leopard, they all have a common ancestor, cat, they all belong to the cat unit. Same way, you can give many examples which says that organisms of a common taxa have a common ancestor. This classification is said to be phylogenetic. I'm sorry. Engler and Prandtl's classification is a wonderful example for phylogenetic classification. Even we take as classification of all organisms into 5 kingdoms in your textbook is also come under phylogenetic system of classification. Now, chromosomal number, look here, chromosomal number and structure is taken as a criteria in cytotaxonomy, and the chemicals like proteins and secondary metabolites are taken as a criteria in chemotaxonomy. These are the words which are there in your textbook which help in phylogenetic classification. One word I missed out here is numerical taxonomy. All the observable characters are given codes and they are fed in the computer. In that way you classify the plants. That is said to be numerical taxonomy. That completes the introduction part to plant kingdom, children. Watch out for some kind of important statements which I have written on the board which can be of useful. Now, we start with alga. Listen, in plant kingdom, if you look at the last 5 years of NEET exam, from which unit of plant kingdom majority of questions have come, there is one unit from where maximum questions have come, and that is alga. So therefore, give more importance to this unit, because time and again at least 1 to 2 questions have come in from alga in every last 5 years. Now, what are the unique features of alga? They are mostly aquatic in nature. They have a thalloid plant body. The cell wall contain hydrocolloids, algin and agar. They can hold plenty of water. Brown alga and red alga, mostly they contain. This is important. It contains all types of chlorophyll. Chlorophyll A, B, C, D. A and B is seen in green alga, A and C in brown alga, and A and D in red alga. And isogamy and oogamy, all 3 are observed. What about from bryophytes to angiosperms? You see only oogamy. This is the only group where all 3 types of sexual reproduction is seen. Haplonitic life cycle is very common, but you also see haplodiplonitic life cycle, and you can also see diplonitic life cycle. Diplonitic is seen in fucus, haplodiplonitic in majority of brown alga and red alga, and in green alga. The majority of alga or in green alga, the life cycle is haplonitic. The sex organs are unicellular. Now, when you look at Spirogyra, if you look, these are the 2 cells. All right, these are the gametes. So this is said to be a sex organ, which is single cell. This is said to be a sex organ with single cell. So therefore, the sex organs are unicellular without any jacket. The female sex organ is oogonium. Don't say archegonium. Archegonium is seen for the first time from bryophytes onwards. The female sex organ is oogonium. External fertilization is very common. Just like in fishes, amphibians, both the gametes are liberated into water and fertilization occur inside water. Different shapes of chloroplast is also seen, not in all alga but in green alga. And most of the carbon dioxide fixed on earth, is because of alga. That's a very important introduction points to alga. I'm pretty sure that you know all. This completes the introduction. Now we go to green alga. Look here, children. There are some side headings which we are going to follow to remember the 3 types of alga. Now, what are the side headings? First, we talk about habitat, then cell wall, then we talk about the pigments, then we talk about the reserve food, then we talk about the chloroplast, then we talk about the flagella, then we talk about vegetative, asexual and sexual reproduction. For all the 3 classes of alga, we'll be following the same timetable. Now, habitat. They are mostly freshwater. I'm not saying always freshwater, but mostly they grow in freshwater, rivers, ponds, lakes, etc. Cell wall, outer pectin, this is important, and inner cellulose. Just like in higher plants, the cell wall is made up of pectocellulose. Pigments include chlorophyll A and B. The reserve for the starch, and it is stored in protein bodies called pyrenoids. Look here, the pyrenoids will have the central protein core. Surrounding it, the starch gets added up. Central protein core, around it, the starch gets added up. So this is said to be pyrenoid. Pyrenoids are present inside the chloroplast, in stroma. Pyrenoids are seen inside the stroma. Means they are not present in the cytoplasm, but they are present inside the chloroplast. This is important. The chloroplasts have diversified shapes. Flagella, this is equally important, 2 to 8, and the apical flagella. Vegetative reproduction is by fragmentation, and you see non-motile or motile spores. Motile spores are called zoospores. They are seen in Chlamydomonas. Coming to sexual reproduction, there may be isogamy, anisogamy, or oogamy. Gametophyte is dominant and thalloid. It produces non-motile isogametes, Spirogyra, or motile isogametes, Ulothrix. Anisogamy is seen in Eudorina, and oogamy is seen in Volvox and Cara. That's an important point. All right, these examples are important, children. Spirogyra, isogametes which are non-motile. Ulothrix, isogametes which are motile. And Eudorina and oogamy. Zygote undergo meiosis. Life cycle is haplonetic. Sporophyte is represented by zygote only. Look here. If you look at one example of green alga and study, this is one filament, and the filament contains many cells like this. Yes or no? Yes. What happens at the time? What happens at the time of sexual reproduction? The plasma membrane and protoplasm, it is going to contract and form gametes. Look, gametes formed. So these gametes are looking alike, hence you call them as isogametes. Look here, both of them show amoeboid movements. When both of them show amoeboid movements in the lateral conjugation canal, you are going to see the formation of a diploid zygote. Same is the case here. Both of them show movements, and in the lateral conjugation canal, somewhere here, you see the formation of diploid zygote. And this zygote undergo reduction division and give rise to spore, and the spore will germinate under favorable conditions into a long filament. So this is gametophyte, haploid, male gametangium, haploid, female gametangium, haploid, male gamete, haploid, female gamete, haploid, zygote, diploid, reduction division to produce spore, haploid. It's very clear. The only diploid structure in its entire life cycle is zygote. So therefore, the life cycle is rightly called haplonetic life cycle. That's a very important point to remember. Children, some of the examples of green alga I have listed here. In fact, you know it. Just go through this. Unicellular protista and SCP chlorella. Unicellular motile protist and also a green alga, Chlamydomonas. Colonial alga with motile cells, remember, and oogamy, Volvox. Filamentous unbranched, Spirogyra and Ulothrix. And isogamy with non-motile gametes, Spirogyra. Isogamy with motile gametes, Ulothrix. And isogamy, Eudorina. Oogamy is Volvox and Cara. And what is also remember is ribbon-shaped chloroplasts are seen in Spirogyra. All right. Girdle-shaped chloroplast, collar-like chloroplast are seen in Ulothrix, and cup-shaped chloroplast is seen in chlorella and Chlamydomonas. This is the point I was talking about. This is the gametophyte, you could see here. And some of the cells are acting as gametangia. Then what happens? These gametes are present inside the gametangia. Gametes fuse to form zygote. And this zygote undergo reduction division and give rise to spore, and spore germinate to give rise to filament. This is the only structure which is diploid. The remaining all are haploid. Hence the life cycle is said to be haplonetic. Well, you know that. Children, it took a little time for us to get rid of green alga, but you know what is the layout we are following? Habitat, cell wall, chloroplasts, pigments, reserved, vegetative, sexual and asexual, followed by some examples. Okay, chalo, we'll go with brown alga next. Habitat, they're mostly marine. Plant body, very important, is actually a sporophyte, not in your book, but remember that. It's a massive plant body, kelp you call. with air bladders. This is important. Ectocarpus does not have holdfast type and frond. It is just filamentous without holdfast type and frond. That point is important. Frond is edible. Look here, you know, in laminaria, you have seen the diagram here. It is how it looks. All right, this is the holdfast, this is type, and this is the frond. You see in all the kelps the structure like this, but not in Ectocarpus. In Ectocarpus, this structure is not there. Cell wall, outer algin and inner cellulose, pigments A and C. Fucoxanthin is going to give rise to brown color. Reserve is laminarian starch and mannitol. This is an alcohol, all right, which act as a what is called reserve. Next, asexual spores are zoospores. Zoospores, very important. Both they are pear-shaped with two lateral unequal flagella. This is how a zoospore look. Pear-shaped with two lateral flagella. Not only zoospore, even the sperms or spermatozoids look like this. So therefore, we say that zoospore and spermatozoids look like this. This life cycle you can understand better when I go with the diagrams. But listen here. This is the table, children, which I want you to remember at any cost, because that makes plant kingdom look very simple. Now look, the same table we are going to apply for all plants, starting from alga to gymnosperms. Now listen carefully. What is the table? So first, sporophyte. What is the meaning of sporophyte? It is a plant which produces spores. Listen carefully. It's a plant which produces spores. So what sporophyte will have? Sporophyll. It contains leaves. These leaves contain bags on it. What is synonym for bag? G-I-U-M. And these bags will have spore mother cells. They undergo reduction division. This is the reduction division and give rise to spores. Up to here, no problem. Now you understood? The job of sporophyte is to produce spores. Listen this carefully. Memorize this properly. Sporophyte, sporophyll, sporangium, spore mother cell, spores. Sporophyte contain leaves called sporophylls. Leaves contain bags called sporangia. Bags contain spore mother cells. Reduction division to give rise to spores. Now let me see whether I can repeat without looking at the board. Sporophyte, sporophyll, sporangium, spore mother cell, spore. Great. All right, just like you, I can remember this properly now. So the job of sporophyte is to produce spore. Now, same way we'll start with gametophyte. Gametophyte, gametophyll, gametangium, gamete mother cells, gametes. You want to say like that. But I want to make a small correction. Don't exactly replicate the cycle similar to sporophyte. Simply say, the job of gametophyte is to produce gametes. Gametes are produced inside bags called gametangia. That's it. No fills. All right, simple. Gametophyte, male gametangium, male gamete, female gametangium, female gamete. And these gametes fuse to form zygote. Zygote generally, not in alga, generally develop into embryo, and embryo will develop into sporophyte. Clear? I repeat the gametophyte again. Let me see whether I can tell the gametophyte. Gametophyte, male gametangium, male gamete, female gametangium, female gamete. Gametes fuse to form zygote, embryo, and embryo I take to sporophyte. Let me see the entire table we have discussed in 2 installments, isn't it? Let me club both of them. Sporophyte, sporophyll, sporangium, spore mother cell, spore. Gametophyte, male gametangium, male gamete, female gametangium, female gamete. Zygote, embryo, and again sporophyte. This is looking fine. Look here. For another half an hour, we are going to see this table. All right, use this table in order to classify all the plants. Let us see, does all the plants behave similar to this table or not? All right, just look at Look at brown alga. Let us see. This is sporophyte, holdfast type and frond. Frond, holdfast type and frond. All this put together. Sporophyte. The frond is green and appear like leaf, hence comparable to sporophyll. On the frond, you see many bags, sporangium. Inside that, you see cells, spore mother cells. Reduction division to give rise to spores. You know that these spores are pear-shaped with two unequal flagella. Spores and germination. Some of them will develop into male gametophyte. Some of them will develop into female gametophyte. What gametophyte should produce? Male gametophyte produce male gametangium. You can even say antheridium, which produce male gametes. And it produce female gametangia. This is female gametophyte. Will have the female gametangium. Don't say archegonium, oogonium. And inside that you see the egg. Both sperms, which are again biflagellated and pear-shaped, just like the spores. This is a very popular question. Both of them, sperm and egg, get liberated. External fertilization. Zygote is formed. No embryo. This zygote again develop into sporophyte. This is looking nice. Even brown alga properly fits into this table. Sporophyte, sporophyll, sporangium, spore mother cell, spores, zoospores. Male gametophyte 50%, 50%. Female gametophyte. This produce male gametangium. This produce female gametangium, oogonium. Male gametes, just like here, pear shape. Female gamete, both of them get liberated, zygote. Brown alga look very simple now. All right, this is life cycle, is haplodiploidic life cycle, unlike green alga, which is mostly haploidic. All right, so now what are the examples, children? This is important. Filamentous simple thallus is the ectocarpus. Is it a kelp? The answer is no, it is not a kelp. The frond is edible. In laminaria and sargassum is a very important question. Diploidic life cycle is seen in fucus. Highly branched frond is seen in dictyota. So the frond has air bladders in all the three kelps, laminaria, fucus and dictyota. Ectocarpus does not have holdfast type and frond. All right, life cycle is haplodiploidic. That looks nice. That completes brown alga, children. Then we go to red alga. All right, they are mostly marine, just like brown alga. They can be seen even in deep waters. This is a very popular question about the cell wall. It contains agar, it contains polysulfate esters, it contains carrageen, it contains mannans, it contains galactans and calcium carbonate. A polymer of mannose is mannans. A polymer of galactose is galactans. This is there in the cell wall chapter of the cell, the unit of life. So the cell wall is highly heterogeneous and contains plenty of phycocolloids. This is one question which was asked maximum number of times in NEET. So be careful, try to remember it properly. Pigments, chlorophyll A and D, and the red color is because of phycoerythrin. This was asked many times. Floridian starch. It resembles glycogen and amylopectin in structure. It resembles glycogen and amylopectin in structure. This is easy. Flagella is absent in spores and gametes. In both spores and gametes, flagella is absent. It is just like, you know, when I'm standing in the signal, when there is a red light, I stop, isn't it? Signal, traffic signal I'm talking about. Red light, stop. Stop. Red alga, they can't move because there are no flagella either in male gametes or in spores. Then in traffic, opposite to red is green, isn't it? Green means go. But in alga, opposite to red is not green, but it is brown. So in brown alga, it is go. It means that both spores and gametes are motile. Whereas in red alga, spores and gametes are non-motile. I think by this time you know all this. Sexual reproduction is mostly oogamous. No question of isogamy and anisogamy here. Only oogamy is seen here. Complex post-fertilization changes. Well, I don't want to discuss in detail about the sexual reproduction in red alga children, that is carpagonium and all that, because that is not a part of our syllabus. But just remember, oogamy and post-fertilization changes are complex. Edible frond is porphyra. Highly branched thallus is polysiphonia. Agar yielding is Gelidium and Gracilaria. This is a very popular question. And all of you know agar is used in tissue culture medium to make the medium semi-solid. Otherwise the egg plant will drown and die because of suffocation. You know that. Carrageen, this was also asked in the previous NEET, is obtained from a plant called chondrus. There are some important statements I've written. It is just like a small worksheet, all right? I don't want to talk. I want to take a small pause. Just run through these lines. Please go through these lines. Both green and brown alga show isogamy, anisogamy, and oogamy. Over 70 species of marine alga are edible. Alga, the major primary producers. Diatoms and dinoflagellates are also alga. All of you know that. They are the major producers, and they are also what is called an example for alga. Diatoms and dinoflagellates. Children, that completes this unit called alga. It took a little time for us, but you are extremely good now, all right, with regard to this alga. Now we go to bryophytes. So what is seen for the first time in bryophytes? First time you see embryo. First time you see archegonium. In brown alga, it was oogonium, not archegonium. First time land habitat. There are some alga which are terrestrial, but bryophytes, all of them, except one or two, all right, like Rixia, all of them are terrestrial. So therefore, the land habitat. And meiosis occur in spore mother cells, not in zygote, as you see in majority of green alga. So first time embryo, I don't think this is a big statement. We'll exclude this. All right. So therefore, you will take this embryo, archegonium, and land habitat. The reason why I have written here is that meiosis occur in spore mother cells in all the members. That is the reason I have written, but again it's confusing. So therefore, let us take these three: embryo, archegonium, and land habitat. What are the unique features? When I say unique, children, it is seen only in bryophytes. In no other group you are going to see this. Sporophyte is physically attached to gametophyte. Sporophyte is physiologically, it is depending, it is absorbing water and salts from gametophyte. Both gametophyte and sporophyte lack vasculature. That is very important, which is seen here. Characters of all types of bryophytes. So in all bryophytes, you're going to see this. Gametophyte is green and independent. Gametophyte is attached to soil. Gametophyte may be thalloid or leafy. What do you see after gametophyte? Gametangium. Gametangium may be on the same gametophyte, mosses, or on two different gametophytes, Markensia, dioecious. Then male gametes are sickle-shaped and biflagellated, and the female gamete is non-motile egg. Water is needed for fertilization. Zooidogamy means the male gametes are motile and they are fusing to form zygote. Embryo is seen for the first time. It develops into sporophyte. The sporophyte do not have sporophyll, but capsule sometimes in mosses is green in color, just like a leaf. It is comparable to sporophyll. And the capsule produces sporangium. The sporangium contains spore mother cells. They undergo reduction division to give rise to spores. And the spores get liberated either by elaters in liverworts or with the help of peristomial teeth, as you see in mosses. All right. Look here, children. If you observe very closely, I have kept all the statements in the same table which I said 5 minutes back. Go back and see. Gametophyte, yes. After gametophyte, gametangium, yes. After gametangium, gametes, yes. then water, zygote, yes, embryo, sporophyte, sporophyll, sporangium, spore mother cell, and spore. And what the spore will give rise to? Again give rise to gametophyte. So this is the table which is looking very simple for you to follow. Let us put this table for this group called Hepaticopsida. First let me show you the diagram which may be helpful to you, then we can read this. Alright, so look here children, this is the diagram. Chalo, let us see this. From where shall we start? You can start from wherever you want. You can start from sporophyte or gametophyte. But in green alga and also in bryophytes, the dominant plant body is gametophyte. So therefore, let us start with gametophyte. Okay? This is gametophyte. Thalloid, looks like the lobes of the liver. This gametophyte contains a long stalk called antheridiophore. And this antheridiophore bears antheridia. And this antheridia is going to bear spermatozoids. So go back to the table. Gametophyte, male gametangium called antheridium, male gametes called spermatozoids. Now I want to go to one more thallus. This is one more Markensia, which contain a long stalk called archegoniophore, which contain only archegonium. Look here, only archegonium. And this archegonium contain an egg cell. So let me go with this. Gametophyte, male gametangium, female gametangium, male gamete, female gamete. That's very simple to remember. Gametophyte, male gametangium, male gamete, female gametangium, female gamete. Female gametangium is called archegonium. Female gamete is egg cell. Male gametangium is antheridium. And the male gametes are spermatozoids, which are biflagellated and sickle-shaped. What will happen next, children? Look at this. The spermatozoids, in the presence of water, fuse with egg cell to form a diploid zygote. Zygote for the first time develop into embryo. And this embryo will develop into a sporophyte, as you are seeing here. Nice diagram. This sporophyte is attached to gametophyte, physically and physiologically dependent on gametophyte. What does this sporophyte contain? Foot, seta, and capsule. What do you see after sporophyte? Sporophyll. Capsule, to some extent is comparable to sporophyll. Inside the capsule, there is a spore sac. The spore sac contains spore mother cells. They undergo reduction division to give rise to spores. 50% of the spores develop into gametophyte with antheridia. The remaining 50% will develop into gametophyte with archegonia. So easy to remember. I repeat again. Gametophyte, gametangium, gametes. Male gametangium, male gametes. Gametophyte, female gametangium, female gamete. Zygote, embryo, sporophyte, sporophyll, sporangium, spore mother cell, reduction division to give rise to spores. Spores, some of them will develop into gametophyte with antheridia. Some of them will develop into gametophyte with archegonia. Let us see what I have written here, children. Now it's more easier for you to follow. Just please go through this. Gametophytes are unisexual or dioecious. You could see it. On one gametophyte, there is only antheridia, and another only archegonia. Same thing I've written. One type of gametophyte produce only antheridia, and other gametophyte produce only archegonia. How is the gametophyte? It appears like the lobes of the liver. That is the reason why it is called liverwort. Then this is an important point. On this thallus, if you observe, on this thallus, all right, you're going to see multicellular green bodies. which are vegetative propagules, and you call them as gemmae. That point is important. And they have unicellular rhizoids. Remember that point also. Then after gametophyte, you know gametangium, male gametangium or sex organ is called antheridium, and it produces biflagellated spermatozoids. The female gametangium is called archegonium, and it produces non-motile egg. Children, gametophyte, male and female gametangium, male and female gamete, zygote, embryo. I told you that we are in the same process. Fertilization occur in the presence of water. Spermatozoids has to move in the presence of water. That is the reason we call them as amphibians of plant kingdom. Zygote develops into embryo. First time you see embryo, and this embryo produces into sporophyte. Now what does the sporophyte possess? Foot, seta and capsule. And it is a total parasite on gametophyte. If it is a liverwort, it is a total parasite because the sporophyte is non-green. But if you go to mosses, the sporophyte, the capsule turns green. So therefore it is depending on gametophyte only for water and minerals. But anyway, in your textbook all this is not mentioned. One thing is clear, sporophyte is physically and physiologically dependent on gametophyte in all bryophytes. Whether it is total or partial is something which depends upon which group of bryophytes we are talking about. But that point is not there in your book, so therefore leave it. Spores are produced from spore mother cells and they get liberated by elaters. Spores directly develop into thalloid gametophyte. Some of them develop into gametophyte with antheridia, and some of them develop into gametophyte with archegonia. Sporophyte is relatively less developed, children. Example is Markensia, Porella and Rixia. Porella is a leafy member. Okay. This is looking simple. Gametophyte, gametangia, gametes. Gametes to fuse to form zygote, embryo, sporophyte. Sporophyte will have sporophyll, sporangium, spore mother cell, reduction division spores. Spores give rise to gametophyte. This is turning out to be easy. Then we talk about mosses, children. Mosses, let me first show you the diagram which will help you to understand. And it's very clear now that we should start with gametophyte. All right, let us see. This is interesting. This is the gametophyte. Early gametophyte called protonema. Filamentous gametophyte called protonema. Juvenile, you can say. This protonema bear many buds. These buds are going to give rise to an adult gametophyte called gametophore. So it's very clear that when it comes to mosses, the gametophyte is dimorphic. What is the meaning of dimorphic? Young gametophyte is filamentous and the adult gametophyte is leafy. That point is important. And what is the meaning of leafy? It contains multicellular rhizoids, root-like structures. It contains a stem-like structure, and it contains leaf-like structures. You may ask me, why are you saying root-like, stem-like and leaf-like? Reasons? That doesn't have stomata, that doesn't have cuticle, that doesn't have xylem, that doesn't have phloem, that doesn't have mesophyll, that doesn't have stele. In spite of all this, structural difference with regard to roots of higher plants, stem of higher plants and leaf of higher plants. Just like root, stem and leaf of higher plants, they does the job of absorption of water, root, conduction of water. Dead parenchyma will help in conduction of water even though xylem is not there. And they also help, leaves-like structure can also help in photosynthesis. Just like a wing of a bird and a wing of an insect, both of them are structurally dissimilar but functionally similar. Similarly, you can say that the vegetative organs of mosses and the vegetative organs of higher plants, they are structurally dissimilar. I told you the structural dissimilarities, but functionally similar. Nothing wrong in calling them as analogous organs, right? Now, with that, children, we completed gametophyte. By this time, you can tell what is there after gametophyte. After gametophyte, you should see gametangium. Listen here. At the tip of the main axis, you see antheridium. At the tip of the lateral branch, you see archegonium. Unlike in Markenshire, where antheridia and archegonia are present on two different gametophytes, hence dioecious or unisexual. Here, antheridia and archegonia, both of them are present on the same gametophyte. Therefore, we say that funaria is bisexual or monoecious. You can say bisexual or monoecious. Now, what do you see inside the archegonium? You see an egg cell. What does antheridium produce? Spermatozoids. They get liberated, they fuse with egg. From here onwards, it is similar to liverworts. Zygote is formed, embryo is formed, the embryo develops into sporophyte, foot, seta and capsule. Capsule is comparable to sporophyll. Inside that you see spore sac. Spore sac contains spore mother cells. They undergo reduction division to give rise to spores. Spores get liberated with the help of peristomial teeth. Elaters in liverworts, peristomial teeth in mosses. And after they get liberated, first they develop into protonema. And this protonema later produces adult gametophyte called gametophore or leafy stage or adult stage. This is looking simple, children, isn't it? All right. So that completes the life cycle of bryophytes. What did you observe here? Gametophytic stage is dominant and free living, and the sporophytic stage is dependent on gametophyte. That is very unique about this group of plants. Then, without wasting any time, we'll talk about the pteridophytes. First time, what is seen in pteridophytes? First time, vascular bundles. Hence, you call them as vascular cryptogams. First time, the dominant stage is sporophyte. What about in alga and bryophytes? The dominant stage is gametophyte. Here, the dominant stage is sporophyte. First time, you see true root, stem and leaves. Why we say that? Because in bryophytes, we see root, stem and leaf-like structures, because not only they are haploid, because they are part of gametophyte, they lack vasculature, stele, collenchyma, sclerenchyma, etc. But here, they are diploid, a part of sporophyte, they contain vasculature, they contain stele. That is the reason we say true root, stem and leaves. First time, the roots are adventitious roots. The stem may be rhizome, as you see in ferns and equisetum. Or the stem may be prostrate and creeping on the soil, as you see in Selaginella. Then coming to leaves, children, this point is important. The leaves may be small, microphyllous, that is seen in Selaginella. The leaves may be large, macrophyllous, that is seen in ferns. This point is equally important. Strobilus. It's a long axis which contains sporophylls. Just like in flowering plants, we say flower is an organ which contains sporophylls. But here these are not flowers. So strobilus, comparable to flower, so which contains sporophylls. All right. And strobilus is seen in two examples, Selaginella and equisetum. You have to remember that. Heterospory, we'll be discussing this in detail, is seen in some members like Selaginella and Salvinia. You see heterospory. Nice. We brought some important lines of your textbook, all right, and we kept here. They are very important. Don't forget them. Then common names of pteridophytes. They are vascular cryptogams, because cryptogams include Thallophyta, Bryophyta and Pteridophyta. In these three, these are the only cryptogams which contain vasculature. First, true land plants. Remember, true. If they ask you first land plants, bryophytes. First, true land plants. First group of plants in which the sporophyte is dominant. Dominant sporophyte. First group of plants with true root, stem and leaves. First, heterosporous plants. Plants with many precursor characters to seed habit. This is important. All right. And what are unique features here? Both sporophyte and gametophyte are independent, green, attached to soil, either with adventitious roots or unicellular rhizoids. Maybe you'll be understanding this once I complete this unit. Mostly homosporous. Example is equisetum and ferns, they are homosporous. But some members are heterosporous. This is again very important. Selaginella and Salvinia. Both microphylls and macrophylls. Feather-like leaves in ferns. Hence, ferns can be used as ornamentals. Stem is mostly underground, rhizome or creeping with rhizophore in Selaginella. So there are 4 classes. Salaeopsida, they are extinct. Salaeotum, Lycopsida, Selaginella, Spinopsida, Equisetum, and Pteropsida, ferns like Pteris, Dryopteris, and Ediantum. Remember these 4 classes. Then we talk about ferns. All right. Characters of ferns, children. Just if you look at the diagram, I think you'll understand this better. Same way, we'll go with it. So this time for a change, we'll start with sporophyte, children. We'll start with sporophyte. Yes, this is the sporophyte, which contain rhizome, which contain feather-like leaves called fronds, which contain adventitious root at its base. What do you see after sporophyte? Sporophyll. This is the leaflet. You have seen ferns many times. When you look from distance, these leaves appear green. In fact, they are green. But when you go near them, there are some leaves which contain brown colored dots on its abaxial side. Don't call them as vegetative leaves. They are similar to vegetative leaves in appearance, but they are no more vegetative leaves. They are called sporophylls. So these is the brown colored dots I said. So after sporophyte, sporophyll, they are present in groups called sori. Outside chance is a typical fern character. Then after sporophyll, you see sporangium. Look here, this is the sporangium, children, bag. Inside that you see spore mother cells. They undergo reduction division to give rise to spores. Spores get liberated. They reach the soil and they develop into a gametophyte. This gametophyte is called prothallus. Prothallus. And this gametophyte bear antheridia and archegonia on it. Antheridia produce spermatozoids, multiciliated, a typical fern character. Archegonia contain an egg cell. Spermatozoids fuse with egg to form a diploid zygote. Zygote develops into embryo. So on this embryo will later develop into a sporophyte. Look here, you could see a sporophyte developing from embryo. And this is a sporophyte. The table is very properly explained here. Sporophyte, sporophyll, sporangium, spore mother cell, spore, gametophyte, male gametangium, female gametangium, male gamete, female gamete, fuse to form zygote, embryo, and sporophyte. Perfect. So it's not that you don't know all this. We are revising all this, and this is the best way to revise it. So if you can remember this table, sporophyte, sporophyll, sporangium, spore mother cell, spore, gametophyte, male and female gametangium, male and female gametes, zygote, embryo, and sporophyte, your job of learning looks more easier. So once you follow a rhythm, children, it's more easier to remember things, right? Then try to compare and contrast the sporophyte and gametophyte in ferns. This is sporophyte and this is gametophyte. I'll tell you the difference. Sporophyte will have true root, stem, and leaves. Gametophyte doesn't have root, stem, and leaf. It is caudate, heart-shaped, and thalloid. Sporophyte contain vasculature. Gametophyte doesn't have any vasculature. Sporophyte contain true roots. Gametophyte contain root-like structures called rhizoids. Sporophyte job is to produce spores. Gametophyte job is to produce gametes. Your textbook says Gametophytes are very sensitive because it doesn't have vasculature. They grow only in moist and shady areas. For that sake, you know what the sporophyte do? Even though it has a license of growing anywhere in the world, the sporophyte picks narrow geographical regions wherever moisture is plenty, because its gametophyte cannot survive in all types of soils. So your textbook says sporophyte of ferns are present in narrow geographical regions because water is needed for fertilization, and added to that, its gametophytes are very sensitive, highly reduced, and lack vasculature. That is the reason the sporophyte is seen in narrow geographical regions. That completes a very important group. Let me see what I've written here. Did I write in the same order? Did I write in the same order? I should have written in the same order. Let us see. Yeah, roots, adventitious roots, correct. Stem, rhizome, correct. Leaves, large feather-like fronds, correct. Sporophyte, represented by root, stem and leaves, correct. Sporophyll, some leaves have brown colored sporangia subtended on it, it is right. Sporangia are bags with spore mother cells, correct. They contain spore mother cells which undergo meiosis, correct. Spores are of same type, homosporic, and get liberated and reach the soil, is correct. Gametophyte, mostly green, independent and thalloid. Prothallus. Rarely the gametophytes are heterotrophic, rarely, but mostly it is green, independent and thalloid. Male gametangium, antheridium. Male gametes are multiciliated spermatozoids. The female gametangium is archegonium, and the female gamete is egg. Fertilization is oogamy in the presence of water. Zygote and embryo develops after fertilization, and that embryo will develop into sporophyte, life cycle. Both gametophyte and sporophyte are independent and green is an important point. This point is equally important, children. Just go through this. Gametophytes are without vasculature, hence cannot grow in all types of soils, and water is needed for fertilization. So ferns look very simple, and you're going to thank whom? Not me, but this table. All right, which is going to help you to understand in a simple way. Sporophyte, sporophyll, sporangium, spore mother cell, spore, gametophyte, male gametangium, female gametangium, male gamete, female gamete, zygote, embryo and sporophyte. I think I'm repeating more than once. It's not needed. You're all very sharp and intelligent students. I should not revise this many times. Example of ferns, children: Pteris, Dryopteris and Erianthum. Even Azolla is used as biofertilizer. Marsilia. This is heterosporous, okay? Salvinia, based on the diagram in your textbook. All right. It gives an impression that there are roots and leaves. But I want to tell you, Salvinia doesn't have any roots. Some of the leaves are dissected and appear root-like. Okay? So, Salvinia is a rootless pteridophyte, just like Ulfia in angiosperm, which is rootless. Then what are the root-like structures in your textbook diagram? They are nothing but dissected leaves. So, the leaves are of two types. This question will not come, all right, of your glassum. Then the most important one, that is Selaginella. Please concentrate here. Selaginella. This is one important example. Concentrate more. Roots that are adventitious. All right, they arise from the base of rhizophore. Stem, it is weak and creeping on the soil. Leaves, they are microphylls. Leaves are microphylls and heterophilic. Why you call them as microphylls? If you look at the stem, children, this is how the stem look like. All right, this is the stem which is creeping on the surface of the soil, and you could see the leaves. They are smaller like this. They are very small. And there are two types of leaves. Look here. This leaf is small and remains small. This leaf is large. So there are two types of leaves on the stem. So nothing wrong in saying heterophilic. Cotton, larkspur, coriandrum, developmental heterophilic. Buttercup, ecological or environmental heterophilic. Selaginella, habitual heterophilic. So it means that two types of leaves is because of its genetic nature, habitual. This is a rhizophore, children, which contain roots at its base. Rhizophore with roots at its base. Does it have strobilus? Yes. I'll show you the diagram. The sporophyte is with root, stem, leaves, megasporophylls. Same way. Look here, the small change. Because this is heterosporous, I use the word micro and mega before the words. Chalo. After sporophyte, sporophyll, I use a prefix mega. Sporangium, I use a prefix mega. Spore mother cell, I use a prefix mega. Then spore, I use a prefix mega. Then female gametophyte, then female gametangium, then female gamete, that is egg. Look here, that's the only change. All right, I repeat again, look here, this is the long strobilus, children. This is the long strobilus, concentrate. All right, what is this? This is a megasporophyll, concentrate, megasporophyll, because it's arising at a nodal region of this strobilus, megasporophyll. What do you see after megasporophyll? Megasporangium. Look here. This is megasporangium. Bag. GIUM means bag. Megasporangium. Then inside that there is one megaspore mother cell. It undergo reduction division and give rise to 4 megaspores. Out of 4 megaspores, 3 megaspores disintegrate. This is the functional megaspore. This is the functional megaspore, because the 3 other megaspores are going to disintegrate. This is the functional megaspore. What will happen to this functional megaspore? This undergo many mitotic divisions. This megaspore undergo many mitotic divisions and develop into many cells. All right, develop into many cells. Few cells on the top transform into gametangium with an egg cell. Look here, I make it more simple. This is the strobilus. Sporophyte will have strobilus. Okay, this is the strobilus. Sporophyte will have megasporophyll, megasporangium, megaspore mother cell, reduction division, 4 megaspores, 3 will disintegrate, functional megaspore. It undergo many mitotic divisions and develop into a female gametophyte, and on the top of it, few cells of it transform into female gametangium called archegonia. Let me see what I've written here. Sporophyte, this is the sporophyte. Megasporophyll, leaves present at the base. This is megasporophyll, okay. Megasporangium, it is present in the axil. This is the axil, megasporangium, correct. Megaspore mother cell, there is only one megaspore mother cell inside. It reduction division. Look here, there is only one megaspore mother cell. It undergo reduction division. 4 megaspores, 3 will disintegrate. 1 is functional. This is the functional megaspore. All right, 4, but only 1 is functional. The female gametophyte. What will happen to the female gametophyte? Megaspore undergo mitosis and produce many cells inside them. Megaspore, free nuclear divisions, many cells. They're all the cells which are produced inside. All right, and this is said to be a female gametophyte. And few cells of the female gametophyte on the top transform into archegonia. And inside that you see an egg nucleus. So easy to remember. The table is same? No. Sporophyte, sporophyll, sporangium, spore mother cell, spore, gametophyte, gametangium, and gamete. Only thing is that we put a prefix mega before that because Selaginella is heterosporic. Megasporophyll, megasporangium, megasporangium, megaspore mother cell, megaspores, 3 disintegrate, functional megaspore, undergo many mitotic divisions, female gametophyte, few cells on the top, archegonium, inside that an egg cell. No problem. Absolutely no problem. This is easy to understand. All right. So the female gametangium is archegonium, inside that you see egg. Tell me for male reproductive system, can you tell? Easily you can tell. Microsporophyll. I use the prefix micro. Then sporangium, microsporangium. Then microspore mother cell. Then microspore. Then male gametophyte. Then male gametangium. Then male gamete. Where all this will be there. On the top, look here. This is the strobilus, no? This is megasporophyll. These are all the sporophylls, no? Somewhere here, this is the microsporophyll. Microsporophyll. And in the axil of it, you see microsporangium. Just like in this axil, you see megasporangium. This is strobilus. Microsporophyll, microsporangium, microspore mother cells, reduction division, give rise to microspores. Microspores develop into male gametophyte. At 13 cell stage, they get liberated. All that is not needed. Male gametophyte produce male gametes. What will happen to the male gametes which get liberated? Male gametes getting liberated. Look here. This is the megasporangium, megaspore mother cell, megaspore, and this is the female gametophyte. We already have seen this diagram, and few cells on the top transform into archegonia. Chalo, look what happens to the male gametes. Listen carefully. Male gametes are going to, this megasporangial wall degenerate. This male gametes fuse with egg to form a diploid zygote. Zygote develops into embryo. Children, this is the statement which you have to remember. Concentrate now what I'm talking here. Where is this strobilus? This strobilus is present on the parent sporophyte. This strobilus is present on the, this is the strobilus. This strobilus is present on the parent plant. On the parent plant, what did you see now? You see the formation of a diploid zygote. On the strobilus, embryo on the strobilus. Ante, the development of embryo, embryogenesis, and the entire embryogenesis is occurring on the parent plant, because this is the parent plant. This is the parent plant on the sporophyte. It means that the embryo is nurtured by the parent sporophyte for a certain period of time. Once this embryo is properly developed, then this entire female gametophyte is thrown out, and this embryo will develop into a daughter plant. Children, I'll tell you 5 characters for any plant to have seeds. 5 characters. What are those 5 characters? Listen. 1, it should be heterosporic. Selaginella shows heterosporic. 2, single functional megaspore mother cell inside megasporangium. Selaginella qualifies here also. 3, single functional megaspore inside megasporangium. Selaginella qualifies here also. 4, which is highlighted in your textbook. Fertilization and embryo development should occur on the parent sporophyte for a certain period of time. Selaginella also qualify even here. Now I want to tell you the last character. Megasporangium should be covered by an integument. Unfortunately, the megasporangium of Selaginella is not having any integument. When it doesn't have any integument, we don't use the word ovule. Megasporangium with an integument is ovule. Selaginella doesn't have ovule because no doubt it has megasporangium, but it doesn't have integument. When there are no ovules, naturally no seeds. Selaginella show many precursor characters to seed habit, but lost the character of seed production because megasporangium is not covered by an integument. That's it. That's about the Salacinella unit, children, which I'm pretty sure that you understood. The only thing what we did is we use a prefix: sporophyte, microsporophyll, megasporophyll, microsporangium, megasporangium, microspore mother cell, megaspore mother cell, microspore, megaspore, male gametophyte, female gametophyte, male gametangium, female gametangium, male gamete, female gamete, zygote, embryo, and again sporophyte. That is the reason why we say Salacinella and Salvinia are heterosporous pteridophytes. Why heterospory? Because there are two types of spores, microspore and megaspore, which we don't see in alga, which we don't see in bryophytes, which we don't see in majority of pteridophytes. Only in Salacinella, Salvinia, Marsilia, Isoetes, and some other 7 to 8 members of pteridophytes, all right, are with heterospory. Even in pteridophytes, majority of them are homosporous. That's an important point to note. All right. Look here, female gametophyte with embryo is liberated and reach the soil and produce sporophyte only after sufficient development on parent plant. Heterospory and embryo development on parent sporophyte for a considerable period of time is a precursor first to seed habit. This is very important. Children, this is the diagram of the sporophyte. This is the strobilus I was talking about. This is megasporophyll, microsporophyll, megasporangium, microsporangium, and all this, you know it. This diagram looks little dull and dim. I'm sorry for that. All right, unique features of Salacinella. Yes, heterophilic. Heterospory, yes. Precursor characters to seed habit, yes. Microphylls, yes. Strobilus like equisetum, yes. Rhizophore, yes. Now coming to equisetum, this is based on the diagram. Roots arise from the base of rhizome. Rhizome is seen. Look here. If you look at the stem, children, you see a wall of branches. Wall of branch on the stem. That is the reason why you call it as horse tail. Right. And the leaves are very small on it. You see a wall of leaves. And the leaves are very small on these branches. Only one type of spores, homosporous. An important example, children. Salacinella and Salvinia, heterosporic. Equisetum and ferns, homosporous. Salacinella and equisetum, strobilus. Salacinella and equisetum, microphylls because the leaves are small. Ferns, macrophylls. And Salacinella and Salvinia, many precursor characters to seed habit. Pteridophytes look ridiculously simple now because you understood the table, and based on the table, we are able to decipher the entire ferns and Salacinella. We are in the last 5 minutes, all right, where we are going to get rid of the last group, children, that is gymnosperms. This was asked maximum number of times. The majority of them are xerophytes, all right, and they show adaptations to dry conditions. What are those? Thick cuticle, sunken stomata, hypostomatous condition, reduced leaf lamina, spinous. All right, this is a very popular question in NEET. Please don't forget that. After habitat, habit. Majority of them are trees, children, and shrubs. Interesting, herbs are not there. So these plants, first of all, doesn't know how to swim. They are not hydrophytes and they are not herbs. They are either woody shrubs or woody trees with trunk. Sequoia, this is the tallest plant in the plant kingdom. Root system is taproot system. This point is important, coralloid roots. They look like corals. They're dichotomously branched, look like corals. And look here, they're negatively geotrophic. And they contain blue-green alga in them, which can fix atmospheric nitrogen. Mycorrhizal roots, well, you know, we already studied in biological classification. association of fungi and pinus. The pinus plants are tall, looks like a coniferous tree. But the problem with that plant is less number of root hairs. So the fungi says, don't worry, I'll stay on roots and absorb water and minerals. In turn, give me some food. And both of them come to an agreement. That is said to be symbiosis or mycorrhiza. So both coralloid roots and mycorrhizal roots are symbiotic. But only coralloid roots fix atmospheric nitrogen, not mycorrhiza. Secondary growth is seen for the first time. There are some pteridophytes like isoetes which can show, but secondary growth cutting across all members is seen for the first time in this gymnosperms. Cycas, the stem is unbranched and pinnately compound leaves. Pinus, the stem is branched with needle-like leaves. Jingo, it's a living fossil. The stem is branched and show palmately lobed simple leaves. This is the third diagram in your NCERT textbook. Sequoia is the tallest. Outside chance for this question to come, neatum. Xylem vessels are present. Leaves, persistent leaf bases, which forms an no, I'm not a big fan, all right, of these statements because these questions rarely come in the exam. All right, Cycas, pinnately compound leaves. Pinus, needle-like leaves called pines. Jingo, palmately lobed simple leaves. This is an important point. Leaf shows many xerophytic adaptations. Coming to reproduction, children. All right. Again, we go in the same order. The plant, sporophyte. Cycas is dioecious. So you see a male plant and female plant separate. All right. So on the male plant, you see the presence of a cone. Cone is comparable to flower. You may ask me, why you are not calling a flower? Because there is no perianth, there are no sepals and petals. So therefore I say cone, male cone. All right, then this is the male cone, children. Imagine at the tip of the male plant, you see a male cone. This is the male cone, which contain many microsporophylls like this. You could see the head. Male cone. So male cone is over. What do you see after male cone? The male cone contain microsporophylls is over. How is the microsporophyll? Let me draw a microsporophyll here. All right. This is how the microsporophyll look like. Yes. Microsporophyll. What does microsporophyll should contain? It contain microsporangia. This is one bag, another bag, another bag. These are microsporangia. What does microsporangia contain? Microspore mother cells. What happens to this microspore mother cells? They undergo reduction division and give rise to microspore. It is covered by outer exine and inner intine. What will happen to microspore? It will develop into male gametophyte. So it will develop into 2-cell structure, sometimes 3-cell structure. This is male gametophyte. And this male gametophyte is liberated, children. Liberated. This is also called pollen grain. It gets liberated. And it is going to reach the ovule. Now after reaching the ovule, again a pollen tube is formed. And one of these cells, generative cell, divide and produce 2 male gametes. Let us see the entire cycle. Sporophyte. The plant body is a sporophyte. This is the sporophyte. All right. Sporophyte. This is the sporophyte. Roots, stem, leaves, pinnately compound leaves, and the cone. This is the sporophyte, male plant. This is the male cone, flower. This is sporophyll, microsporophyll, sporangium, microsporangium, spore mother cells, microspore mother cells, spores. Microspores are pollen grains, male gametophyte, no male gametangium, no antheridium. Directly you see male gametes. After liberation, these pollen grains reach the ovule by wind pollination. After reaching the ovule, they produce a pollen tube, siphonogamy, and one of the cell will develop into produce male gametes. Same thing I have written here. The male cone contains many microsporophylls. Microsporophylls give rise to microspore mother cells, meiosis. Microspore develop into pollen grains. Pollen grains are liberated 3-cell stage. In Pinus, they are winged and liberated 4-cell stage. Pollination occurs by wind. What happens next? Pollination is direct as pollen grain reach the ovule without stigma. The male gametophyte develops a pollen tube after reaching the ovule. This is called siphonogamy. The pollen tube carries the male gametes for short distance. Then male gametes are released from pollen tube near the archegonium. After release, the male gametes move independently towards the egg. So in cycas, I'm talking about cycas. In cycas, not only siphonogamy, but also zooidogamy, because the pollen tube doesn't go up to the archegonium. The pollen tube get liberated just above the neck of archegonium into archegonial chamber. So the pollen tube liberates the sperms into archegonial chamber, and in the archegonial chamber, the sperms fuse by themselves with egg. The male gametes are large, motile, multiflagellated in cycas and gingo. When the remaining gymnosperms, the male gametes are non-motile. Cycads exhibit both siphonogamy and zooidogamy. All right, other members exhibit only siphonogamy. So what is important, children? Dioecious condition, terminal male cone, 3-cell pollen release, cycas, I'm talking about cycas, wind pollination, direct pollination because pollen grains are directly reaching the ovule, not reaching the stigma. Presence of both siphonogamy and zooidogamy. And this is important, largest male gametes in plant kingdom, cycas shows. The female gametophyte, if you look, the female sporophyte bears megasporophylls. Megasporophylls are loosely arranged. So therefore we use the word lax. We don't use the word strobilus. And megasporophylls, let me show you the diagram of megasporophyll. These are loosely arranged, and this is megasporophyll, children. And these are the megasporangia, very important. Look here, these bags are naked, you can see them. Naked ovules, very typical of gymnosperms. All right. So megasporophyll bear megasporangia on the lateral side. They are exposed and open. Naked ovule. This point is important. And within each megasporangium, children, from here onwards, it's very simple. Now this is megasporangium. It is covered by an integument. Think about angiosperms. Nucellus. One of the nucellar cell develop into megaspore mother cell. Reduction division. 4 megaspores. Just like in Selaginella. Out of 4 megaspores, listen, 3 megaspores disintegrate. 1 functional megaspore. This undergo many mitotic divisions and develop into a female gametophyte. You can even call it as endosperm. Few cells on the top transform into archegonia. So let me show you the entire female reproductive system. The female plant with a lax, megasporophyll, megasporangium with an integument ovule, megaspore mother cell, megaspore. This is the functional megaspore which develop into female gametophyte, female gametangium called archegonia. And all of you know, children, the pollen tube reaches here. These are the sperms. Sperms fuse with egg to form a diploid zygote, an embryo. So it is no more an ovule now. It is now called a seed. Seeds get liberated. So the same thing I've written here. Megasporangia, megasporophyll bear megasporangia on the lateral side. Within the megasporangium, nucellus differentiate into megaspore mother cell. Megaspore mother cell undergo reduction, division, and give rise to megaspores, 4. Out of 4, only 1 is functional. The functional megaspore undergo repeated mitotic divisions, look here. And that results in the female gametophyte called endosperm. Endosperm is a pre-fertilized product. It is haploid. It is highly reduced gametophyte. It is parasitic on sporophyte. You may ask me, endosperm is a nutritive tissue. It becomes nutritive only after fertilization, not before fertilization. Endosperm is parasitic on sporophyte. That's a very popular question. Gametophytes are highly reduced and parasitic on sporophytes in gymnosperms and angiosperms. It was asked many, many times. All right. The female gametophyte differentiate into archegonium. 2 to many archegonia are present inside a single ovule. Each archegonium contains a single egg cell, and the egg cell is also the largest in plant kingdom. And key features: no female cone or lax, not true female cone, lax is seen. Ovules are exposed, endosperm is haploid, pre-fertilized product. After fertilization, ovule develop into seed. The zygote is formed inside the archegonium. Seeds often show polyembryony. Why? Because there are 2 to 8 embryos, 2 to 8 archegonia. You could see 2 to 8 embryos. The zygote undergo free nuclear division. It develops into proembryo. Suspensor pushes the developing embryo deep into the endosperm. The embryo differentiates into radical, plumule, cotyledons. And the seeds of gymnosperms represent 3 generations. Seed coat, parent or present sporophyte, embryo future sporophyte, endosperm is parent or present gametophyte. Seeds in pinus are called Chilgoza seeds. So here I have written at the end, children, some few important points. Antheridia, archegonia, oogonia, true root, stem and leaves, embryo, vascular tissues, cryptogams, phanerogams, flowering plants, water needed for fertilization, water needed for pollination, water not required for fertilization, zooidogamy, male gametes motile, siphonogamy, wind pollination only, many pollinating agents including wind, precursor to seed habit, but not seed, precursor to seed habit along with seed, all precursor characters. Seed plants are spermatophytes, naked ovules or naked seeds. Ovules present. Megasporangium is seen. Some heterosporous pteridophytes also. Heterospory. Dominant gametophyte. Dominant sporophyte. Non-vascular sporophyte. Vascular sporophyte. It was little marathon class, children, in this plant kingdom which we did. But I'm pretty sure that we covered every line which is important for your textbook. Do revise this one more time. Read your textbook. I hope that this is very much understandable to you. We'll come with one more chapter tomorrow. Till then, keep reading. Thank you.